Thanks for joining us. We're excited today to have kind of a special session for our Aquatic Invasive Species webinar series with three presenters, and it's also a special extended session. So we're going for an hour and a half this time instead of our typical hour. This is a special session on starry stonewort and science, prevention, and management. So we have three speakers here. We have Dan Larkin and Nick Phelps from the University of Minnesota and Ryan Wersal from Minnesota State University, Mankato. And each kind of have a set of slides that they'll be sharing. There will be a question answer session that we'll facilitate through a tool called Slido. You can go to slido.com and enter the code STARRY to get to it, and it works great. You can upvote questions that you'd really like to hear the answer to, and you can put questions in there at any point. If for some reason you're having troubles with Slido or would just rather not use it and you put your question into the chat, we have Michelle here, and Michelle is going to help transfer everything over there. So your question won't get lost if you happen to put it in the Zoom chat for whatever reason. We do have everyone muted for the duration of the webinar. We have quite a few folks on the line. So just go ahead and use Slido for that, or if you're having any problems, you should be able to chat the hosts and panelists, and Michelle, who's behind that AIS Detectors program handle, will help you out with whatever issue you're having. And I think with that, I'm going to kick it straight over to our presenters. I think Dan Larkin is starting things off. So Dan, I'll let you do an introduction and get started with your slides.
Great, thanks, Megan. It looks like maybe when you, oh, there we go. All right, so I'm going to assume everything is presenting fine and people can hear me. Thank you all for coming today. I'm excited to present and participate in the Q&A and also to hear what Ryan and Nick have to say. For my portion of the presentation, I'm going to address the following questions. What is starry stonewort? How has it spread in North America and Minnesota? What are some key characteristics of its ecology and impacts? And what so far have been sort of the general outcomes of management efforts? I want to start out by acknowledging our funding sources and the various entities here at the U that have been involved in the work that I'll be presenting today. And there's really too many people and too many collaborators to point out by name individually, but I've listed a few of the people who've worked in my lab who have particularly contributed to what I'll be presenting today. And then we have a lot of agency collaborators from Minnesota, Wisconsin, and Indiana, Departments of Natural Resources, local units of government, Blue Water Science, just to highlight a few. So I always like to start talks on starry stonewort by defining what it is, and I like to start with the tree of life. So these are the domains of living organisms here on Earth, and the reason I do that is because yes, starry stonewort is an algae. It's a green algae. It lives right here, sister to plants. But these stars also mark other groups that have what are called algae. So it's important to realize that algae is more of an ecological or behavioral term. It's not an evolutionary term, and it brings a lot of baggage to the discussion because people have these associations with things like harmful blue-green algae, which are actually bacteria and couldn't be more distantly related to starry stonewort. Zooming in a little bit, it's specifically a type of green algae called a charophyte that is closely related to the land plants. And the things we typically think of as plants are vascular plants. And so I think it's important on the one hand to recognize starry stonewort as an alga. It's not a vascular plant, and plants' vasculature or veins are something we can exploit, for example, in chemical treatment, and we don't have that opportunity with starry stonewort. I think that'll be relevant to some of what Ryan talks about. But it's also important to recognize it as basically a plant. Structurally, it's a macrophyte. It's not anything like a filamentous algae or phytoplankton. Starry stonewort is native to Europe and Asia, and it was first found in North America in the mid-1970s in the St. Lawrence River. And it's dioecious. That means males and females are separate individuals. And only males have been found in North America to date. There was a report out of Canada of female starry stonewort, but I think that was pretty convincingly refuted in the paper that I cite here. Spread in North America, therefore, is by fragments and baubles. It's clonal or vegetative spread, not through sexual reproduction. This is a zoomed-in image of a bauble. They are star-shaped, asexual reproductive structures that give it the name starry stonewort. And boater movement is the presumed driver of spread. And Alex Badges, who's a quantitative ecologist with MACERC, I think has shown that pretty convincingly through an analysis he did recently, looking at expansion of starry stonewort within lakes and showing that the entry point has been near accesses. In its native range, starry stonewort is a rare species. In fact, it's a red-listed species in many countries. That means it has a conservation status as threatened or endangered and is a conservation priority. So, for example, Paraphyte experts in Europe it's really surprising to them that it's being treated here and behaving as an invasive. But it is indeed spreading more or less within kind of the Great Lakes footprint in the US and Canada. This map from USGS, each of these points represents populations of starry stonewort with larger, darker points representing more populations. And this is what that accumulation has looked like over time since the mid-1970s, with Minnesota highlighted there. It was first found here in 2015. It wasn't really on people's radar much before about 2010. And so that rapid accumulation after 2010 is some combination of continued spread and it actually going and being found in new places, but also just much more awareness of starry stonewort and a lot more work on it that has led to identifications of probably some of which were already existing populations before that time. This is what the history looks like within Minnesota. That first point was Lake Coronis in central Minnesota. And you can see as we've picked up more populations over time each year, it's sort of remained as a hub in central Minnesota and a hub in northern Minnesota, with that southeasternmost point being the one example we know of starry stonewort being found in the Twin Cities metro. So one of the things that we were really curious about and wanted to get a better handle on was the ecology of starry stonewort and its potential impacts, specifically to native aquatic plant communities. And so Carly Wagner, who's pictured here, did a master's with me. Carly is now with the Minnesota DNR. And she did dive-based transect sampling in 3 lakes in the state to look at relationships between starry stonewort and native plants. She did really extensive sampling in the summer of 2018. And what she found is that as you had higher starry stonewort cover, that was associated with reduced native plant abundance and reduced native diversity. So here, this is showing that abundance relationship with starry stonewort cover on the x-axis. As that increases, you have a decrease in native cover across all 3 of these lakes. And then in terms of native species richness, that's just the count of the number of native species that were found in a given quadrat. And you can see that also had a shallower but significant decline across these 3 lakes as you got more starry stonewort. Now that's just one snapshot in time and starts to give us a glimpse into patterns of relationships. But what's beneficial is to look at the same locations over time, over multiple years, and see how the plant communities are changing. So I'm just going to orient you to a few of these animated plots. The red is starry stonewort abundance. The teal is native species richness. And you can see in these transects in Lake Coronis, over time, you saw a big increase in starry stonewort cover and a decline in native species richness. And you can see across all these transects, that was a pretty strong and consistent pattern of this shift towards starry stonewort dominance and loss of native species. It was more of variability in outcomes on Moose Lake. On the left here, I've highlighted a couple of transects that show that sort of similar pattern of increasing starry stonewort, decreasing native species. But on the right, we've got some where the story was a bit more complicated, and where we actually, in some cases, saw sort of a retraction of starry stonewort, contraction of starry stonewort, and some recovery of native species. So that was based on 3 years of data. One additional year was sampled thanks to Ranjan Muthukrishnan. And with the addition of that 4th year on Coronis, in a lot of transects, the pattern I had shown you before held, with that 4th year still showing dominance by starry stonewort. But then there were other cases where we saw something in that 4th year, we saw a contraction there. And so I guess my takeaway here is that starry stonewort can become dominant quickly, but it can also recede quickly. And so the longer term effects here are going to depend on what's that trajectory of starry stonewort abundance over time, how much resilience is there in these native plant species. So while for repeated dive sampling of the same transects 4 years is a good amount of time, the big questions are, 5, 10, 20 years, what's going to happen? So Carly's work highlights a lot of interannual variability in starry stonewort abundance. And that leads us to another kind of time-based question, which is, what about seasonal growth patterns, which we call phenology? This has practical implications. If there's a high degree of temporal overlap with how native macrophytes are growing, that could potentially lead to greater impacts. If they're kind of trying to do the same thing at the same time, there could be more, kind of sharp elbows between the species. And then growth patterns are also relevant to treatment timing, which again, Ryan will talk more about. But here's some photos from our sampling. Here's Mike Verhoeven with starry stonewort sampled from a single point. In the summer, you can see just a really extraordinary amount of biomass. And then the picture on the bottom right, I think, was from February, sampling under ice, and you can see starry stonewort still green against a backdrop of brown carol. So we did 2 years of sampling in 2 lakes, again, Coronis and Moose, because we wanted to kind of have a climate gradient, some difference in climate between those 2 locations. And these figures are showing the accumulation of starry stonewort biomass over the season for 2 years, and in 2018 for 2 lakes. And what stands out to me is there's a good amount of interannual variability with the peaks differing between the 2 years, but a good amount of actually geographic similarity between at least Moose and Coronis, showing similar patterns in 2018. What's going on with the baubles is a really important question. And so we did sample baubles. There was very high spatial and temporal variability in bauble density. You can see these are really messy data. And this is something Ryan has investigated as well with his students. If I squint, I can kind of imagine a story of baubles decreasing through the spring as they're sprouting and turning into new plants. And I can imagine bauble production or recharge happening as those matured plants are able to dedicate energy and resources to forming new baubles that will then live in the sediment and regrow the following spring. This pattern is a distinctly later phenology than common native macrophytes. So in this figure, starry stonewort is the kind of filled teal. And we've got other native charophytes are that first peak, and then other native vascular plants are that second peak that's highlighted. So you can see it's shifted later. And if we look at some other submersed invasive species, two that we focus on a lot in Minnesota, curly leaf pondweed is at first peak, Eurasian water milfoil is at second peak. We again see that starry stonewort is really sort of doing something different with this persistence of biomass later into the season than these species. I think that sets up a bit of a conundrum for control. So when should we treat for high effectiveness with minimal collateral damage? Early on, growth of starry stonewort is fairly minimal and regrowth is likely. But in mid-season, by the time starry stonewort biomass is really high, native species are peaking, so we have to worry more about non-target impacts. Later on, it's sort of just starry stonewort by itself, but by that time, the bulbal bank is probably already recharged, and you're nearing the time when it's going to senesce on its own. So I sort of question, how much value for the next year you're going to get by sort of kicking it when it's down at that point. Okay, so turning to management for the final section of my presentation, there's been a lot of work by a lot of parties in Great Lakes states on starry stonewort management. There's been valuable contributions of small-scale field and lab trials that have been published that have gotten at management outcomes and effectiveness, but we lacked a broader synthesis to evaluate management effectiveness in general, sort of, the state of the practice as it stands today. So in a project that Wes Glisson did a tremendous amount of work on with funding from MACERC, we worked with state DNRs and lake management contractors to get all available treatment and survey data from Minnesota, Wisconsin, and Indiana. And here's what is happening out there. There are about 400 total treatments in this time period from 2010 to 2019. The dominant strategy was use of algicide, mostly copper-based algicides, but also non-copper herbicides as well. And then there were a handful of these other approaches, physical techniques like hand pulling or mechanical harvest, diver-assisted suction harvesting, etc. So we had a lot of survey data thanks to the monitoring that had been done by agencies and contractors. And so we had sort of three types of data. We had whole lake point intercept surveys. This is very commonly used in evaluating aquatic plant management. A grid of points throughout the littoral zone of a lake are sampled, and you get information on the distribution of a target species. Of course, for starry stonewort, we're not doing whole lake treatments. So narrowing down on where the treatment is actually happening, we have targeted sub-PI surveys that are taken within treated areas. And then in a few cases, we have sort of these case studies where, for example, there can be a design where you look at a treatment response and compare it to what's happening in an untreated reference area nearby, or where there's biomass sampling, which gives us kind of richer information on starry stonewort abundance than just sort of its distribution in points across the lake. Along this continuum, we sort of go from the highest spatial scale and the most available data to the finest spatial scale and the least available data. So for whole lake surveys, we had data from 35 lakes over a decade, over 150 total PI surveys. And we get information on frequency of occurrence, and that's just the proportion of occupied points. So if these green stars are starry stonewort, it's that proportion of points out of the whole survey. We also analyze rate density, and that's in the paper that'll be forthcoming soon, but I'm not going to present those data today. So I'm going to show you a figure that is going to, for each lake, show the years since starry stonewort was first found and its lake-wide frequency of occurrence. So what proportion of points, which could be 0 to 100% of the sampled points. And this is what that data looks like. Each of these lines is a different lake, and we can see this general pattern of starry stonewort continuing to expand across these lakes over time. So the management at this scale has not been effective enough to stop expansion once you get these larger populations. But I'll also point out that the conclusions here are limited by lack of good baseline data. And that's because monitoring of untreated lakes over time has been rare. And we really need that as a point of comparison. You know, what would happen without treatment is not as well characterized. So drilling down to these targeted sub-PI surveys, I'm going to show before and after data. Again, each line is a different lake before and after treatment. That thick black line is the overall mean response, which is a relatively small but significant decrease in starry stonewort abundance with treatment. But what that's obscuring is what's really going on, which is in some cases we're getting good treatment outcomes, starry stonewort is decreasing, but in other cases there's no change, or in a couple, starry stonewort even increased despite management. So general pattern of treatment decreasing extent of starry stonewort, but outcomes highly variable. I think what's hidden in here is opportunity to identify the factors that are affecting success and try to standardize toward those practices. Lastly, what is really valuable information is when we have what's called a BACI design, where treatments are being compared to an untreated reference area over time. And then we can make stronger inferences about the effects of treatment itself. These are relatively rare data, but Lake Coronis, for example, has been a hotbed of these really high quality data being collected. And we can see in this case, there's 3 lines here, that dashed line that sort of stays up there as an untreated reference area. And then there's 2 treatment areas, one that got algicide treatment, one that had mechanical harvesting and algicide. And you can see in both of those cases, there was really good reduction of biomass. I'm going to close by talking about hand pulling. Early detection really expands the treatment menu for starry stonewort. And if you find the populations when they're small enough, hand pooling with or without algicide treatment can be a highly effective approach. These are data from 3 lakes in Minnesota, sampled by Minnesota DNR and Blue Water Science, where those groups went out and did careful hand pulling with trained divers. And what these data are showing is the amount of starry stonewort biomass that was removed from each of these hand pulling events. And you can see that over time, they're getting less and less and are sort of starting to chase scraps of starry stonewort. And the hope is that maybe with sufficient sustained effort, these populations could even be eliminated. So early detection is a great investment. Starry Track, which is a program of AIS Detectors and MACERC, is an example. Our partners in Wisconsin have AIS Snapshot Day. And then monitoring by, in Minnesota DNR staff, local units of government has led to a lot of the finds of starry stonewort. But the value of early detection is in enabling rapid response. So these local groups, a lake association, for example, where starry stonewort is found, they need support to manage these populations, technical, financial, permitting, et cetera. And I will stop sharing and turn it over to Ryan.
All right. Well, I just wanted to say thank you to the University of Minnesota for inviting me to be a part of this webinar, and thank you all for spending your afternoon to listen to us. So I'm going to kind of pick up where Dan left off. I'm not going to talk a lot about starry stonewort biology and ecology. There'll be a little bit of talk about biology and ecology just mixed in with how I design some of my management. And then I'm going to focus kind of a lot on mechanical harvesting and especially on chemical control, which is a big part of my research and a part of my background as a weed scientist. So just to start, aquatic plant management is more than just trying to go out and kill plants, or in this case, starry stonewort, right? We really have to understand the basic plant biology and ecology, right? So what makes it tick? What is making or causing it to spread? What's causing range expansion? What's causing it to put down bulbils or to flower or set seed, right? So spread and distribution a lot of times is determined by many factors, right? And I have this little illustration up there to denote that. And effective management will also be determined by a number of these same factors, right? We can't effectively manage a plant if we don't really understand how it's growing or how it's reproducing. Okay, we can't effectively manage a plant if we don't understand the site-specific characteristics in the lake. And not every location within a lake is going to respond the same to every management technique, right? There's real, changes in how water flows through a lake, even in lakes that don't have inlets and outlets. And then the other piece that is kind of out there but never really talked about is the public socioeconomic part of it, right? So public perception has a significant role in shaping management programs and the ultimate outcome of management programs based on the information that they receive or don't receive, and then, kind of who they convey their interests or maybe their lack of approval to, right? So we've got to have all of these pieces come together if we're really going to effectively manage a plant. And before we even get to management, we have to have somebody take ownership of the project, right? So a resource manager, stakeholder groups, lake associations working in concert with states or federal agencies, whoever that is, we really need to first, decide, well, who is affected, who's going to take ownership of the program, and then what are the expectations, right? Are we looking at eradication or are we looking at nuisance relief? Okay, because those expectations, if set up on the front end, can really guide management and determine what really is effective or not effective management, right? So we have to lay out the expectations on the front end before we go into actually pulling the trigger and managing. Okay, so when should we manage, right? So are the invasive plants, in this case, starry stonewort, are they impacting the environment? Okay, are they impacting native species? As Dan pointed out, oftentimes it does. Okay, is it impacting recreation? Is it impacting water conveyance? Is it impacting water uptake, right? So is it having some impact on the environment or human use of that water body? Okay, and then, compared to that, what is the real impact of management, right? Should we manage, or will the management cause more harm than leaving it alone? Okay, so there's that balancing act. What techniques are available, and should we use those techniques? Okay, so, manage, no management, no management can be a decision, and there's consequences to that. So having an understanding of that going into a management program is also very important. Right? And as Dan alluded to, understanding life history trends, understanding the phenology, right? So which environmental factors are key in triggering significant stages within starry stonewort, right? So at least in Lake Coronis, in the study that I have up here, we found that water pH and light availability was very important, driving a lot of the seasonal life history trends that we saw in starry stonewort, and especially for timing of bulbul production, right? If we're going to be successful at controlling starry stonewort, we're going to have to figure out a way to address bulbul production. Why bulbils? Because bulbils themselves, the little tiny star-shaped bulbils that Dan showed, they can store up to 73% starch. I mean, that's phenomenal considering all of the other species that are out there, and specifically things that I've studied over my career, right? Eurasian water milfoil stores between 20 to 40% starch in its root crowns. All right, so we've got something that's that small that's storing that much energy. Okay, so not only do they store a whole lot of energy, they make a whole lot of them, right? So in the study we did on Lake Coronis, on average, we were seeing about 1,500 bulbils to 157,000 bulbils per meter squared. And, this piggybacks on what Dan said, but just about the spatial variability in bulbul productions, right? We had some areas where we hardly found any bulbils, and we found some areas with our core sampler that we had 5, 6, 7,000 bulbils in our little coring device. Hey, and based on these numbers for one lake, we estimated annually that they're producing about 4.9 billion bulbils, littoral zone wide, or a potential maximum of 509 billion bulbils, right? So, I mean, so that's worst case scenario. And there's other things that are driving that. But Starry stonewort is a bulbil making machine. And the fact that they store that much energy, it can, play into maybe some longevity in the sediment. And that's one question that we don't have an answer to is how long can those bulbils live in the sediment? Management timing, what I have here would be optimal timing for management. But again, Dan has pointed out that starry stonewort really doesn't behave like a curly pondweed or a Eurasian water milfoil. So trying to take advantage of managing a weaker life stage in this plant is going to be kind of difficult, especially based on our growing season here in Minnesota and across the northern tier United States, and just when this species typically is at its densest growth versus when native species are dominant as well. So now specifically with management, we have to select our management practices so that they correspond to what each individual site is giving us. All right. So there's going to be some site specific, what do I want to say, site specific evaluations that have to be done on the front end so that we know that we're choosing the right tactic to meet that goal, right? So if we have a lot of water exchange, using a pesticide that needs a longer concentration exposure time may not be the best choice. Okay. We need to put all the tools in the toolbox and then choose the tools that are going to give us the most consistent result for that specific place within a lake. And as abundance changes, we also have to be ready to change our management strategy. Okay, so if we're going into this with a management plan, that management plan should be adaptive, and we should be able to change pretty rapidly based on the response of starry stonewort and then localized annual changes within the lake. Okay. So in beginning management programs, I usually recommend starting at a small scale, right? Either at the aquarium scale or at the mesocosm scale, because there's a lot of questions that we can answer just, basic efficacy, formulation differences, non-target effects before we even go into the field. When we talk about pesticide formulations, there's a pretty wide range of products out there, even within aquatics, right? So different active ingredients can be made using active ingredient formulations can be made using different ingredients, right? So 2,4-D is not necessarily the same with every formulation, right? We have 2,4-D, we have 2,4-D acid formulations, 2,4-D amine formulations, 2,4-D ester formulations, and they're all going to perform differently when released into the environment under different water quality scenarios. So what I have up there on the screen, right? So formulations that have different physical chemical properties, just small tweaks in the formulation is going to change how that molecule behaves in the environment and if it can get into the plant. So just that small change in the carbon and hydrogen, you either get no uptake by foliage, and in fact, that particular herbicide molecule is applied to the soil and has to be incorporated in, whereas the bottom one, if we just tweak that formulation a little bit, then we get really good foliar uptake, right? So we can't really understand those nuances until we do a lot of those small scale screening type projects. With respect to starry stonewort, copper formulations tend to work the best, okay? Q-Treme Plus, Captain, right? The ethanolamine complexes work really well. Ethylene diamine complexes like Harpoon or Captain XTR, they can work as well, but I think it's going to be site specific, and a lot of the copper formulations, there's some water quality things that play in there as well. Non-copper results, things like flumioxazin, carfentrazone ethyl, the PPO inhibitors, they didn't seem to work very well. Endothol can work. I found that the monopotassium salt formulation of endothol tends to work better than the dipotassium salt. And actually, diquat in the smaller scale trials was probably the best non-copper alternative that I found. So anything that had diquat or a diquat endothol mix worked pretty well. Okay, so that was small scale. As we scale up to small plot evaluations, right, looking at treated plots versus non-treated plots over several years, we had copper, we had diquat, we had copper plus diquat, looking at some combination treatments. And what we saw there was copper was effective for in-season control. And I think this is one of those important things, is that looking at these small plots, when we went out, when we managed in season, we saw some really good phthalate reduction. Okay, when we went out a year later, all of those reductions were largely erased. Okay, there was a lot of resiliency in starry stonewort, and that we could not maintain that level of reduction year to year. And those were those data that Dan pointed out just a little bit ago with, what's really going on with starry. Well, in some plots we had a good decline, some plots we didn't have a decline, some plots we had an increase. So there's a lot of variability in how this species is responding and what works, what doesn't work. And then there's that spatial, right? Not every lake is responding similarly to management. So copper was really good, reduced above ground biomass every time. In the field, diquat was not effective. We could not get diquat to work by itself. And in the first series, the first set of trials, bulbal production was not impacted. So we could reduce phthalate biomass, above ground biomass, but we could not affect bulbal production. And that's a big, big problem when trying to manage the species. If we can't reduce bulbals, then it really doesn't make a bit of difference if we reduce phthalate biomass. Okay? In the next set of trials, copper, again, was effective. Copper plus diquat was also effective, right? So we had a combination treatment. The thing with the combination treatment, could we squeeze a few extra percentage of control? Yes. Is it probably going to increase the cost of the application? Yes. Are we going to look at more non-target injury? Probably. But again, in this next series of small plot combinations or copper, we still did not get efficacy in bulbal reduction. And the one takeaway here that I think, I've said this before, but copper efficacy, if we look at phytoplankton, okay, there's a density-dependent relationship. And I think that density-dependent relationship is also going to transfer to starry stonewort, in that as we're treating more and more biomass of starry stonewort, so we have more and more cells of starry stonewort, it's going to require more molecules of copper to impact the more cells. The problem with this, and this is where that density-dependent relationship comes in, is that from a FIFRA, a federal registration perspective, we're limited on how much copper we can put out in a single application and across a year. Okay, so as starry stonewort biomass increases, we come to a limitation on how much copper we can use. So some of that increase or non-impact of management with copper algicides might be due to this density-dependent relationship. And as I alluded to before, the other thing that might be holding us back, not so much from a copper perspective, but in looking at things like endothol and diquat, when we're looking at herbicides, concentration exposure time relationships are vitally important, right? So in the case of Coronis, when we use diquat, most of our diquat was gone in less than 2 hours, which would indicate that there was some significant water movement in and around most of that lake. We lost half of our diquat by 1 hour after treatment and 98% of our dicot by 6 hours after treatment. So we had a very, very, very short half-life of dicot in these plots, which would be the, the overriding reasons of why dicot did not work in Lake Coronis, right? So if we're going to use tools like dicot or endothol, or if we find another herbicide that might work on starry stonewort, we have to remember that we're treating the water around the plants, and we have to keep that concentration around the plants high enough and long enough in order to allow that plant to take it up in a high enough concentration to elicit a lethal response. All right. If we don't meet that CET exposure time, we're going to end up in that no efficacy range. Right. So this is where formulation chemistry can help overcome water exchange, liquids versus granules, adjuvants versus no adjuvants, whatever we can do to increase the contact time in order to get that lethal response is going to be important. Okay. So another part of my research over the last several years is looking at what Dan and Wes had found on Lake Coronis in their observation of lower bulbal densities after mechanical pulling and copper treatment. Okay, so is there something behind that, right? Can mechanical harvesting really work? Because as I just said, it doesn't look like copper algicides are impacting bulbal production on a large scale. So we looked at mechanical harvesters and we scaled it down under controlled settings. So we looked at simulating mechanical harvesting of cutting starry stonewort mass up to about 1.8 meters, right? So we had a non-treated reference. We had clipping once per growing season, clipping every other month. So basically bi-monthly clipping, and then clipping every month, so assuming like a May, June, July, August type growing season. So for 4 clippings total. And we did this based on water depth in Coronis and what Dan and Wes in their study had outlined as far as where the starry was growing. Okay. And what we found is that, we had some seasonal variability. We had studies done in the summer, and then we did some greenhouse studies in the winter, and clipping worked, right? Every time we clipped, we could remove starry stonewort above ground biomass, right? Go figure. Okay. But what was kind of, I guess, surprising is that we could also maintain, at least in these small scale trials, we could maintain a suppression in biomass out to 52 weeks after treatment. Okay, so we were maintaining some longer term biomass suppression. Okay. On average, in these treatments, we were removing about 30 to 40% of the biomass on every clip, right? So every successional clip, we're removing biomass. But I guess the one thing too we have to point out here is that every successive clip, mechanical harvesting became more inefficient because we were removing less and less biomass, right? So that's just one thing to take into consideration. But the big surprise here, or I guess the takeaway here, is that clipping did reduce bulbal density. And the more that you clipped, the more that reduction became significant, right? So at 52 weeks after treatment, we saw a significant reduction in bulbal densities if we could clip starry stonewort 4 times. Okay, so what does that look like? So if we go back to life histories, if we were just removing, 22 to 88% of the bulbal density, right? We go from 1,500 bulbils down to 184 bulbils per meter squared, or 1,500 bulbils down to 1,100 bulbils per meter squared. And then if we go on the larger end of that, but we're still removing bulbils, we're still preventing starry stonewort from putting more bulbils down. Okay, so could it be? I think harvesting could be a piece of an integrated pest management plan, right? So most harvesters can remove plants at about 1 to 4 acres a day. So if we have 100 acres of starry stonewort, we're going to take about 25 to 100 days in order to cut it. So are we going to use harvesting across, a large infestation like on Coronas? Probably not. But could we use it? Could we make it work in smaller, high traffic areas? I think we could. Could we use it on lake access points? Yeah, around swimming areas? Probably. Okay, so I think we need some more information and some more data on what that would look like in a field-based setting, an operational management program, because again, this was in greenhouses and we were simulating harvesting. Okay, so the more we cut, the better opportunities we have, and the more bulbs, the more reduction we can in bulb production. All right. Now, where we can simulate this, as Dan had pointed out, what the DNR has been doing is looking at hand harvesting. And as part of an early detection rapid response program, I think at least the plots that I'm aware of here in Minnesota and what Christine and her group has done, they have done a really, really good job at getting in there, starting to pull early, and they have maintained that significant level of reduction for multiple seasons where they are just chasing a few scattered plants, and they've really kept those populations down and prevented them from spreading to other lakes. Another technique that may be going operational, maybe some other states are using it. I'm hoping once Dan and his crew are done evaluating diver operated suction harvesting, or DASH, we'll see more and more of it in use. I think it can be highly selective, and I think it can be highly impactful, especially in areas where you've got dense infestations. You can go right for the biomass, you can go right down into controlling those bulbils, because you're going to take those bulbils right out of the sediment. You remove the bulbils, you remove any capacity for that plant to revegetate that area, and long term you're going to reduce that year-on-year recruitment. The other thing I think we might look at, especially in limited areas like around boat ramps or docks or small high traffic areas, might be benthic barriers. Okay, and I know we don't currently permit them in Minnesota, but I know the DNR, I think, is looking at some burlap kind of sheets to see how effective they might be. Right, it's not going to be a selective option, but I think in large traffic areas where you may not want the presence of vegetation, this might be a viable option, not only to kill the above ground biomass, but if you don't have above ground biomass there, you're not going to have any bulbils there. And any bulbils that sprout under the mat, the plant is going to die. Okay, so something to think about. And if there's other folks from other states that are using benthic barriers, if you have any data and are willing to share, I think those of us on this call would be appreciative of any insights you have on the use of benthic barriers for starry stonewort control. So conclusions, right? So if we can only treat or only do something once a year, I think the goal is to maintain the current level of starry stonewort and prevent it from leaving the water bodies that it's in as best we can. I think we're going to be looking at a scenario where multiple treatments are necessary, especially since it's going to be very difficult to pinpoint, that optimal timing. Right now, I don't think there's a silver bullet for starry stonewort. I think it's going to be a long-term concerted effort. Harvesting is going to work on small acreage. I think DASH is going to work on small acreage. And I think, we really need to move to collecting data on what an integrated pest management program is going to look like and what sorts of tools can we use and where can we use them. Prevention and spread, right? The boater check stations, the U of M volunteer ID training, Starry Trek, I think are all very good tools. The more eyes we have out there looking for this, we can get into those EDRR type frameworks. We got to be really proactive. In-lake, preventing in-lake spread or preventing from moving from one lake to the other. Right now, I think we have copper algicides, hand pulling, and mechanical harvesting. I think those are going to be our most effective tools at the moment until we find some new pools of money or some new science or some new techniques that we can evaluate. At the end of the day, I think we need to focus on bulbils. If we're not effectively reducing bulbil production, we just can't win against starry stonewort. I mean, that's going to be that piece in the armor that we have to go after and exploit. So we need additional management techniques, right? We need some other chemicals. We need some more tools. We need an integrated weed management approach. Okay. We need some data on that density dependent, that mass balance approach for copper, right? Are we seeing failures because we just can't put enough copper out there? Okay. What does repeated treatments look like? What is the space and time, right? What should we leave? So there's a lot of work left to do, and it'd be nice to see some of this come to fruition. Okay. So long term, bulbils, 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 and we need to find ways that are cost effective and that we can manage large populations in a cost effective and environmental neutral way. So with that, I'll just acknowledge funding sources: Minnesota Department of Natural Resources, Great Lakes Restoration Initiative, the Minnesota Trust Fund, the LCCMR, Army Corps of Engineers, APCRAP program, and Minnesota State University Mankato. Staff currently and formerly with the Minnesota DNR, several graduate students, and a small army of undergraduates in my lab. So with that, I'll turn it over to Nick.
All right. So thanks everyone for being here, and Dan and Ryan for setting this up. I think you've done a great job sort of justifying the impact of starry stonewort on our lakes, why it's important to think about management. And that last couple slides there, Ryan set it up well for thinking about early detection and prevention as important strategies in the big management puzzle. So those are the two topics I'll focus on for the next little bit here. And I see we're running tight on time. I want to save plenty of it for questions. So I'm going to zip through this pretty fast. The way I've organized this session, or this section of the talk, is around some of the frequently asked questions in the research to support my answers to them. Okay, so the two biggest questions with regards to early detection and prevention are these. And I'm going to first talk about the early detection bit. It's a complicated world out there. Those of you on the call from Minnesota totally understand that. And those of you elsewhere, it's not all that different. We've got a lot of lakes, a lot of boats, and a lot to take care of, and not nearly enough people or resources to cover the ground. So we've spent the last 8 years or so thinking about ways to more strategically look for starry stonewort and other invasive species. And the first question we set out to answer was, how do these species spread? Now, back in 2020, epidemiology and the use of network models was all the rage. Think about these sort of network examples here. The one on the left, a highly connected network with lots of people connected to lots of other people. And on the right, more of the isolation strategy where things are separate. Now, in this case, if you're introducing a virus into the network, you can just imagine how much faster it would spread in the left-hand side. We used the same type of approach to simulate the connection of Minnesota lakes through boat movement and natural waterways. Long story short on this topic, we have a highly, highly connected network in Minnesota. We do not see these sort of sub-networks where these lakes are clustered separate from everything else. Lakes are connected by boats and water, the two primary risk factors for the spread of starry stonewort. So we introduced Starry here in Minnesota, and just the opportunity for spread is very high. The next question to think about, not only can it get there, but if it does, will it survive? And we've spent quite a bit of time, and we're still working on refining these approaches of environmental suitability models. These models consider the environmental factors that might be more or less appropriate for a species' survival. In this case, starry stonewort, we've done projections with a variety of different methods. You can see these maps here. There's a lot going on, and the colors are different across them. The main thing I want to emphasize, though, is that many of Minnesota lakes are very suitable for starry stonewort. Not all the same, and some lakes are, of course, better than others, just like some lakes are good walleye lakes and some aren't. The same is true for Starry stonewort, but much of the state could be considered at risk if the species is introduced. We've looked at future climate scenarios too, and it introduces a whole nother layer of uncertainty. I think it's safe to say, though, that the future is unclear on how this species will continue to establish itself in Minnesota lakes. Now, these models are interesting sort of academic things, these slides I've just shown you in the papers, citations underneath them, but it's not all that usable. So we've spent the last few years trying to take these big, complicated mathematical models and put it online in ways that folks can use it. And so I'll point you in the direction, if you're not already familiar with the AIS Explorer. This is an online tool that does a lot, and I'm going to highlight it a few times here in this in the next few minutes. But the first aspect that I want to show you is an application that takes these network models and the suitability, combines it with other data to estimate the likelihood of introduction and establishment for all lakes in Minnesota. You can see here on the left-hand side, you can select Starry stonewort as one of the focal species. You can look at a variety of different categories and selections on the side, your geographic range of interest. This map that you see is what it looks like. And if you click on any individual lake, it'll give you the probability that that lake will become invaded 5 years from now. Now, these are estimates, of course, built on these models, but they're working quite well, as you'll see in just a second. For those of you who are curious, if you do click on a lake, like let's say Lake Bemidji here, it'll pop up a network of other lakes that it's connected to and how those connections might influence risk. So you'll see here Lake Bemidji is connected, it's a very local network. The lakes that's the most often connected lakes are in fairly close proximity. 55% of the outgoing boats from Lake Bemidji are going to uninfested lakes. For example, if I was a manager up there in Hubbard County, I would be pretty focused on Plantagenet, highly connected to Lake Bemidji, Leech Lake, Cass, a lot of incoming boats from currently infested lakes. So it's just a way to use this tool to prioritize surveillance strategies at the landscape level. Now, for better or worse, these models are being tested in real time. This is a figure from a paper we published a couple years ago. The analysis was done back in 2019. And we had forecasted at the time how many lakes in Minnesota would become infested with Starry stonewort. Now you can see these projections with those error bars of uncertainty. And the little bursts that I just added to the figure are what actually happened. So as we learn from these infestations, we've realized that the model is doing a very good job at predicting the number of new infestations each year. But there's been mixed results on which lakes exactly are most likely to be invaded. This last year, for example, in Crow Wing County, Middle Cullen Lake was number 13 out of a couple hundred lakes in that county for likelihood of infestation. Sort of in that middle tier, maybe not in the top 10, but whitefish, that chain became infested later last year, and it was number 1 on the list. So there's mixed results, but they're useful, and we'll continue to learn and update these models in real time as new infestations happen. Now, another common question is, so we just talked about sort of the lake level, the landscape level, but within lakes, where do we look? How do we prioritize our surveillance and survey methods to increase the likelihood that we'll find it? And a long story short, in the paper cited here, if you're curious to dig in more, there's a few factors that dramatically increase the probability of Starry stonewort occupancy or the presence of this species within the lake. The first is high fetch. That's sort of the distance to the furthest shoreline that is influences wave action. Shallow water and proximity to boat access, both private and public, increase dramatically the likelihood that Starry stonewort will be first found. Over time, we see that starry stonewort will spread throughout lakes and pop up here and there. This is really important to understand for a couple reasons. One, it shows that the boat ramps seem to be sort of a primary introduction point, but it isn't something necessarily about the boat ramp or the environment around the boat ramp that makes it most suitable. Starry can survive wherever else it might want to live in the lake, but that introduction point is important. And as you've heard both from Dan and Ryan, this reinforces boats as a primary vector of spread. So a couple of quick takeaways for the early detection bit. There are risk-based strategies to look between lakes and within lakes, and I highly recommend you think about that if you're strapped for resources. Those of you that are doing surveys out there, it's really important that those data are being shared, both obviously the positive infestations, but the negative results too, because that can guide our models where the species isn't present and somebody has looked. And I promise Megan didn't tell us all to give a plug to the AIS Detectors program, but their participant training and volunteer opportunities are highly valuable, with something like a quarter of Minnesota's starry stonewort infestations being found by members of the public participating in this event. And there's gaps, of course, on early detection. We need more sensitive early detection methods, and some of those, like eDNA, are being developed. And we need a more coordinated, in my opinion, statewide professional surveillance program to complement the public surveillance that's happening right now. Now, really quickly, I'm going to shift gears into the prevention side. Now, we see pictures like this of somebody pulling up a big clump of starry stonewort, and it freaks us out a little bit, understandably. But there's a lot of it down there. And I've heard people sort of bring up the manifestation of a swamp thing. Like this is just coming out of the water and trying to spread its way around between Minnesota lakes. The reality is what does move around is often quite small, and there are opportunities, as you'll see in the following slides, for some effective prevention. These are more realistic bits that might end up on a trailer that's moving between lakes. And Dan and his group did some desiccation trials to see how long really do these different sizes of fragments and bubbles survive outside of the water. This is an important piece to understand risk and the duration of the boat movement and over land. So you can see here from these numbers, it's not all that long for small fragments, and that's what might most likely get missed if someone's doing boat cleaning or an inspection. Bubbles a little bit longer, larger clumps, things that stay wet increase the probability of viability a fair bit. So the key here, like with many of our recommendations on prevention, clean, drain, dry is important for starry stonewort. Now, we've also looked the last couple of years at the effectiveness of various prevention strategies. We looked at both, we look at boaters and then also professional folks, watercraft inspectors, and those doing hot water decontaminations. I'm not going to go into the background of this study. I'm just going to show you some summarized results here. There's a lot to unpack, and I'd point you in the direction of this paper if you're curious to know more. But in general, and this included, I should point out, zebra mussels, aquatic plants, residual water, and spiny water flea as sort of staged experiments with these different participant groups. But boaters were about 56% effective at removing the invasive species. In other words, they removed 56% of the invasive species from the boat. And you can see that number goes way up for inspectors, and it's about the same for decon folks. Now, the boaters in particular, because I imagine that's a population of interest for this audience, that's a pretty good rate here at 56%, but those are folks who actually participated. This was a staged experiment. They were invited to actually do something. And we know there's a massive gap in boater intent and actual behavior. So there's a lot to unpack there in that box alone on how do we shift active participation in prevention activities for boaters to achieve this 56% target. And then there's, of course, ways to make that even better. And we showed clearly that the longer a boater spends doing a boat cleaning, the more invasive species they remove. While that's intuitive, it's a dramatic increase, something like twice as effective from 2 minutes to 5 minutes. Plants were better, if you're curious, than other invasive species that were staged. The inspectors and decon folks, almost 100% of the plants were effectively removed. Again, reinforcing the value of prevention and the effectiveness of it. We have a tool on the AIS Explorer that is an optimization model. This will help you locate watercraft inspectors in the landscape to have the best bang for your buck in sort of intercepting the risky boaters. And you can select Starry stonewort as a focal species or several others if you're curious to dig into that. And I know I'm running out of time, so I'll zip through these last couple slides, but we know, like what I've shown you so far, all these models have been built on an assumption of a status quo, that what we're doing now will sort of continue on into the future and spread will be consistent. We know that prevention, though, will affect the likelihood of spread. If you put a watercraft inspector on a boat ramp and it reduces risk by 80%, that will have benefit. And so we've really dug into the trade-offs here of management investment and how it's allocated to reduce risk on the landscape. So there's really this cost-benefit question. And if we don't do anything, 50 risky boats in this hypothetical scenario go to an uninfested lake. No action's an action, and this has consequences, but costs $0, at least in direct management expense. Now, let's say we implement an inspection program. We spend $200. We intercept 20% of the boats, 80% effectiveness. We can reduce the risk by 16%. Now, these are just hypothetical numbers here that I'm just trying to convey sort of the concept. But doing this for all lakes with our model data is a mathematically challenging problem. But we've spent a long time thinking about this and making it useful for all of you. And that's the most recent addition to the AIS Explorer in the tab we called Intervention Impact. Now you can see, you can select Starry stonewort as one of 2 focal species. You can move across these tabs. In the middle, the choose lakes, effort, effectiveness. We have it all seeded with default data or results based on the empirical evidence, but you can change it however you want. So if you think the watercraft inspectors are more or less effective, by all means, change it within this tool. And what it will send you at the end is a report that estimates both the costs and the benefits of whatever management strategy you put into the system. Now, this allows us for the first time to play around with hypothetical scenarios in the safe space of a computer model. And it doesn't capture the complexity of everything, but it is a data point in your decision-making process. So a few quick prevention takeaways. I encourage all of us to stay optimistic. It's clear that for species like Starry stonewort, reasonable effort can slow the spread of the species. And perhaps we're seeing that with sort of a gradual increase and not a dramatic one, at least at this stage in its infestation in Minnesota. I think there's a lot of value in investing in education strategies that are targeted on behavior change of boaters and also on inspectors at infested lakes. A containment strategy at this stage of an infestation could have a lot of return on investment. And I think there's a lot of room for optimizing signage for boaters at accesses, redesign boat ramps to allow boaters as sort of a dedicated place to clean. These are all things that boaters tell us through our research would help improve their compliance. And there's a lot to do here on prevention still, even though we know a lot. We need to understand and address this intent action gap. And I think there's a lot we could do on coordinating better our prevention strategies across jurisdictions, because of course the species doesn't know political boundaries, and boats are moving all over the place. And a few final thoughts, sort of summarizing my takeaway from all three of these presentations. It's clear since 2015, at least here in Minnesota, when Starry stonewort was first confirmed and Dan came on board, Ryan joined the state a little later. MASERC has invested a lot. Managers have learned a lot. There's plenty at our disposal to take action now. There's no reason to delay implementation of prevention and management strategies with the best available science. There's clear benefit of doing something, like cleaning stations at infested lakes. We know that prevention helps, and I urge us to sort of work in that direction and not wait for the next research paper to come out. With that said, though, we still have a lot to learn. All three of us that have spoke today have highlighted knowledge gaps, ways to optimize our approach, spend money more efficiently and effectively. Research takes time, and while we work on those strategies and work with managers to test and scale them, I urge your patience and support to stick with it and remain vigilant as we work towards our shared goal. And to that end, we must work together. Probably goes without saying, you're all here to learn and be part of this, but there's a lot we could continue to do and build coordinated strategies to identify research needs, co-develop management plans, prioritize locations, all of that at local, state, tribal, federal, academic levels. It's a bit cliche, but all hands on deck is definitely appropriate in this scenario. That's all I have, Megan, and hopefully we've got time for Q&A.
Awesome. Thank you, Nick. I'm going to take over screen share real quick and we can get the Q&A going. Again, we're using a tool called Slido for Q&A. There's already a bunch of questions in there. You can either scan that QR code or go to slido.com and enter the code STARRY. You can upvote questions that you want to see answered or enter your own in. I'll leave this screen up for a little bit and then I'll shut it down so you don't have to just stare at a QR code the whole time we're chatting. But I'll get started and then the three of you can kind of sort out who's going to handle the questions as they come in. The first question is wondering if there's any advice on native plant restoration for areas that are treated for starry stonewort, especially if biomass has been significantly reduced.
I can take a stab at this. This is, kind of an open question in general, following invasive aquatic plant treatment in lakes and other systems, certainly prairies, other terrestrial habitats, wetlands. There's been a lot of work on revegetation methods and good development of species for use. In restoration, that's for submersed plants, that are growing below the water. That is really in its infancy, and there's a lot of interest and effort, a lot of activity around the Twin Cities metro for sure on revegetation methods. And MASERC is supporting an ongoing project in my lab looking at that's focused on aquatic revegetation for preventing reinvasion. After starry stonewort treatment specifically, I don't have any specific recommendations, but I would say the data that Carly collected showed, pretty good capacity for native plants to recover on their own, where starry stonewort biomass had died back. And, a lot of the lakes that starry stonewort is found in Minnesota are pretty high quality lakes with decent water clarity, decent species diversity. So, passive or spontaneous recovery of native plants could probably go a long way, but I think we'll be learning a lot more in the years to come about how we can deliberately reintroduce native species.
Great. Thanks, Dan. Okay. The next question is wondering a little bit about what we know about like source population. So specifically here, they're asking about like that northern and more central population of starry stonewort, and if we know if they've spread from each other or if they might have like a different DNA fingerprint across those two.
I'll take a stab at this one too. This is not something we've worked on here, but at New York Botanical Gardens, Ken Carroll and Robin Sleeth studied this. And they found basically that we're dealing with pretty much a single population in North America from the lakes that they sampled. There was a tiny bit of genetic variation suggesting there could have been either a couple introductions from Europe or mutations that arose here, but they had, I think, 7 samples from Minnesota lakes, and they were all the same, and those were essentially the same as populations of Wisconsin, Michigan, and the Northeast.
Right, perfect. Wondering about the DASH system. Do the divers need certain certifications? And if so, is there a list somewhere for that? And in the same question, wondering what happens to the hand harvested starry stonewort? What does it get where does it get disposed, and what's the regulatory climate around that?
You want to take that one, Ryan, or should I answer?
Well, I think we could probably team this one. As far as certification, I'm guessing they have to be scuba certified in order to dive. I mean, just dive plan and that. As far as disposal, there's going to be some differences from county to county and probably local municipality as far as what they'll take, when they'll take it, and then restrictions on setback distances on how far the disposal place has to be from bodies of water or connected bodies of water. Dan, I know you're working on a project right now with Dash, so you're in there working with the applicator, so to speak.
Yeah, well, really, we're partnering with Kate Haigston and Rainey White from the Leech Lake Band Division of Resource Management, and they've been doing DASH work, and we've been helping a bit with monitoring that. We're hoping in a future webinar to hear from Rainey about their DASH efforts. But it seems like just purely anecdotally, it seems like they're getting some pretty good results now that they've worked out some kind of kinks with the equipment. I will say on the hand pulling, I'm a big fan of it, but I'm not the person who's actually done it, and I know it's really difficult. And so I think it is really important that the people doing the hand pulling are well trained and really know what they're looking for. It's difficult working in water. There's visibility issues that pop up pretty quickly, and you really need somebody who's got that strong eye for identification to seek out these little bits of starry and go after that and leave the native plants in place. So and the cases that I know of in Minnesota that I highlighted, that Ryan highlighted, they've been done by highly qualified divers with a lot of expertise in aquatic plants. And I think that's been the key to their success.
Great. Thanks to both of you. And I'll add, I see Chris Jurek sent a note to us in chat as well from Minnesota DNR. So if you need a permit for disposal, there's a link for that. And I'm going to ask Michelle if you're able to grab that from the chat to the hosts and maybe throw that link into the chat so everyone can see it too. That would be lovely. Thanks, Michelle. All right, on to the next question. Next person is wondering about, besides the displacement of native plant species in some cases, does starry stonewort have other negative ecological effects?
Well, I mean, I've studied biology and ecology of it, but there's other colleagues out there, Doug Pullman from over in Michigan, and he had some data, of some changing in the benthos, that soil water interface and some of the physical chemical properties underneath that mat, sags and dissolved oxygen, changes in pH, those types of things, which can then in turn impact community composition, not only for native plants, but also for other invertebrates, fish, and other species that are inhabiting those areas. And I think Dan probably, when Carly was doing her work, probably observed some of those changes as she went through her transects through the Starry beds.
Yeah, yeah, and I'll also point out, add a couple other things to what Ryan said. There's been research in Canada. There was a PhD student there, Tyler Harrow Lyle, who published some papers looking at other impacts of starry stonewort, including on macroinvertebrates and kind of food web composition in starry invaded lakes. And then in addition to the ecological impacts, as Ryan mentioned, certainly it can be a nuisance, in terms of its growth, filling of the water column, the amount of biomass. So there are also recreational impacts to lake users that are another area of concern.
All right. I'm going to ask this next question, and then I'm going to stop my screen share. So folks might see your screen rearranged just a little bit so you're prepared for that. But I'll also put the link to Slido in chat again for anyone who doesn't have it. Next question is wondering about if mechanical harvest can contribute to spread. So in particular, wondering if there's been successful spread by fragmentation when mechanical harvesting is happening.
I don't know that there's been a study documenting. I mean, undoubtedly there's fragments produced, okay, and there's going to be things that escape. In my experience, starry stonewort's pretty fragile, so it's pretty easy to damage the cells. So I would imagine, and again, this is anecdotal too, that many of those fragments that may escape are probably damaged to the point where they're not viable, or viability is greatly reduced. But I would then offer, there's a chance for fragmentation and establishment and local spread. But if what we've seen small scale holds true, I think the fact that we can potentially reduce bulbal production might outweigh the chance of spread and limited spread through fragmentation. Because if we can reduce bulbals, we can reduce the year-on-year recruitment. If we're just reducing thalli biomass, we're not impacting bulb production. This thing has shown a propensity to produce bulbals pretty much all season long. So I guess I'll leave that. I'll take a reduction in bulbal production with the chance of some fragments.
Yep, and I really like what Ryan highlighted as a use case for mechanical harvesting in terms of, channels coming from an access. So, you can have fragmentation without a mechanical harvester if you don't manage it at all, by boat traffic moving through. And so I think there could be a lot of benefits from using that approach more broadly, where there is thick starry stonewort, where there's a lot of boat traffic, of doing mechanical harvesting there to reduce spread risk. And that is something that Cronus Lake Association has focused on as well. And I think there the benefits would certainly outweigh the risks. You're muted, Megan.
Sorry, thanks, Dan. Do any of you have any thoughts on native plant distribution or frequency data for highly managed systems like those that where copper is being repeatedly applied? They're mostly interested in like Cara or Nitella trends there.
Well, Chris Jurek, I know now, is on the call, and that's something that she's looked at. And Jake Walsh as well, and I think they have a paper out on that that's been published pretty recently. So I won't claim to know what they found, but perhaps Chris would be willing to take follow-up questions from folks about that.
Yeah, Chris, if you have a link that you want to share, you can send it to me, and either Michelle or I will get it into the chat for all to see as well. So then does starry stonewort always grow submerged and rooted? And does it stay close to the lake sediment, kind of where it starts to grow, or will it reach the water surface and start to form mats?
It can get, I've seen it essentially fill the water surface. It's not a mat former in the way that, say, Eurasian water milfoil is, but where growth conditions are best for it, it can get really tall, and you can have, these thick pillows that pretty much fill the water column, but it will start from the sediment. It's not rooted, but it's shallowly attached, superficially attached by rhizoids. In some places, we've seen it really stay short and low in the water column. Other places, it gets thick and lush and can get pretty high up, and that's probably, dependent on nutrients, maybe pH or other factors like Ryan pointed to.
All right. And then thinking about detection methods, has there been any work on using eDNA for starry stonewort detection?
Encourage Dan or Ryan to jump in if there's something Starry specific. I don't believe so. However, we do have a project here on campus looking at developing and optimizing eDNA strategies for plants. And within that plant, broadly defined category, Starry's included. So they have control samples, and this next field season, they aim to go out to lakes and do some validation of their strategy. In theory, it's possible, but they want to double check.
Great. So stay tuned. Maybe we have another future webinar ahead of us. All right. So next question is wondering about noting that early detection being so effective and being able to manage starry stonewort. What are your thoughts on the best ways to educate large numbers of lakeshore property owners on identification?
Megan might have some thoughts on this.
Yeah.
I hate to toot our own horn, but that's something that we work on a lot within AIS Extension, me, Megan, and Michelle, with support from Extension as well as from MACERC. And so we focus on starry stonewort in our AIS Detectors program, which trains volunteers and professionals on early detection of target AIS, not just plants, but animals as well. And then we have, as I think a few of us mentioned, our Starry Trek event, where we have people search. I think this last year is maybe on the order of 250 accesses around the state of Minnesota, and Wisconsin is doing that as well through their own program. So I think it's not that hard to learn how to identify starry stonewort, and there are a lot of great experts in the state who can confirm identifications. And so I think there is a lot of value there in those educational opportunities for, lakeshore property owners, lake users, the public in general.
Awesome. Thanks, Dan. Yeah, and I did not plant that question in, so thank you to whoever asked it. If you three are okay with it, there's one more question I'd like to ask. We're right at 2:30. Is that okay? I see nodding. Okay. So, oops, and I just cleared it out. The question was wondering if you could talk more about using algicides for treatment. Here we go. Can you talk more about algicides that are used, and can it be used on things like flowing systems, or are there restrictions for lake only? And are there impacts to native macrophytes too?
Well, there's a lot there in that question. So as far as algicides that are approved for use in aquatic habitats, we have copper and we have sodium peroxide, basically peroxide. Okay. And I don't know that anybody's really looked at the peroxide chemistries. I don't know that by label we could put enough into the water to get the response that we're looking for. So we're kind of, I guess, looking at copper as the active ingredient. And then within the copper, there's a number of registrations, and then there's a number of formulations within the copper. And those formulations start one with the base copper metal, right? So we have copper hydroxide, we have copper carbonate, we have copper sulfate. So whatever that base copper formulation is, that kind of dictates how the formulation is built on top of that. And then within that, we have different chelators, right? So we have monoethanolamine, triethanolamine, copper citrate, gluconate, copper with the limonene in there. So we've got all of these things that can be added into the formulation to change how it reacts when it's released into the environment. But at the end of the day, it's the copper metal that's doing the killing of the plant. So most of the labels for copper have aquatic habitats on there. And a lot of them are labeled for use in flowing water. There's a number of registrations that are labeled for use in irrigation canals and water conveyance canals, and they use them out west. Most of the registrations have potable water language on them, so they have been evaluated and they are approved for use in drinking water scenarios. So copper algicides are, have been studied since the 60s. They've been around for a while, and they've got a lot of uses built into their labels. Now, that being said, you got to read the labels of the formulations that they're using, and which is why we rely heavily on the certified pesticide applicators in the state to stay up to date with that and actually do the applications and help folks choose the most effective formulation that they think is going to work in the lakes that they're working on.
Awesome. Thanks for taking that last question, Ryan. So with that, it's 2:33. So I'm just going to throw up our final slide here. I want to say thank you to Dan and Nick and Ryan. We really appreciate the three of you coming and sharing your expertise on this topic. We are recording this, so you can find the recording on our YouTube channel, which is sharing on the screen now. We transcribe everything, so give us a week or two, but it'll be up and you should get an email when that's available as well. There should be also a survey that'll pop up as you leave. We always appreciate if you are able to take some time to give us your thoughts on the webinar. And finally, I do have the email addresses here for our three presenters. There were a number of questions that we weren't able to get to. So if you have some lingering questions, hopefully you can reach out to one of them and get the answers that you're still looking for. If you have questions for us about the webinar series in general or Starry Trek or the other programs that Dan and the other presenters had mentioned, you can reach out to us at AIS Detectors at umn.edu. But otherwise, thanks to all of you for joining us today, and we'll hope to see you on a future webinar.
Great. Thanks, Megan, for having us, and thank you, everybody, for attending.
Thanks, y'all.
Great. Thank you.